shouldn't be too long. We're just gonna. I looked ahead, and I think we can actually wrap up not today. A day off tomorrow.
Oh, really?
So
The internet connection was bad.
I can finally take my nap.
we can.
At Yushen.
You gotta do your bio IA.
That was more what I was thinking. Is
Oh yeah, I forgot about IA.
some time to do bio IA. Yeah.
God, just remind me.
So we'll try to wrap this up today, and then we'll take tomorrow off. So and then we'll meet. HLs will meet on Friday, as per usual. Okay.
Wait, so no lesson,
SL.
like no SL? Wait.
No,
what?
we should be able to finish today. 2.9.
It should be.
Wait,
maybe.
so
Oh my god, the IA is still on Friday, man.
I can be on
Chachas.
tomorrow. Like I can be here if you want help with the IA. How about this? Let's make tomorrow's class optional. I'll just start a Zoom. If you have questions about the IA, you can ask them, and I'll just be here for an hour, same time as usual. But it is not required to be there. But the assumption is you are working on your IA.
Oh my chachas.
Okay. So I'll be here. But let's start, okay? So today, I need to start share screen. Okay, today's stuff is found here. Nothing really new, same PPT, same questions. Oops, I forgot to change the question numbers. I'll do that after this. And then a link to a virtual lab. Okay, it looks very similar to the cell respiration virtual lab. This one is a little bit better. It's a little clearer anyway. However, I will, a bit of a warning is that it straddles SL and HL content. So SLs, be a little careful that there might be some things in there that go a bit over your head. So just, if you haven't heard it mentioned in our lessons, then it's probably not something you need to know. HLs, if you check it out and you haven't heard it yet, it's probably because you are about to hear it in the coming week.
Oh dear.
Yeah. Okay.
During this period, I've become so good at not sneezing. Just now I almost sneezed and I just automatically stopped it.
Oh man. Okay. So as I mentioned last time, we would have normally done this experiment where we extract some pigment from a leaf, and then
Potato.
No, not potato, like a real plant. Usually
No, potato.
we use like spinach or something. I forget what we use usually. Anyway, some kind of green plant. And then we do what's called thin layer chromatography. And what this does is it takes a very strong solvent and it separates the green color that you can, like the very dark green color that you can extract out of a leaf. And because the color of a leaf is actually a mixture of many different types of pigment, like not just chlorophyll, there might be some other types. What will happen is the pigments will like get separated from each other, and then based on their size and their molecular composition, they will move along this plate. That's what the white thing is here. Sometimes you can use filter paper, but it's not very accurate. We use like this slide with sort of this white like silica thing on it anyway. So it moves along, kind of like gel electrophoresis. So it moves along, and then based on how big the molecule is, it will stop at different levels along the plate. And then we can identify it based on like the distance that it traveled, and we can name the different types of pigment. So one of the activities in here is that. So you'll see there's two different experiments. There's chromatography and there's photosynthesis. So the chromatography one, if you look through here, shows exactly how it works. So again, go through this on your own just to see how the experiment would be done if we were there. And it shows you exactly, this is like the setup that we would do in class. Okay, so with this one uses filter paper.
So if
Can
you-
we do something similar before?
We would, and I can actually show you a video of a different class that's done it. Can you see this okay?
Yes.
So this is a
Yeah.
time lapse. This is a time lapse of
is that
the experiment happening, okay?
long hair, skinny, female.
So you can see they make like a dark stain of color from a leaf, and you can see as the solvent runs through, it pulls apart the different types of pigments.
Wait, what? How do we get that dark ink?
So what we would do is you would take a leaf, and essentially, I'll play it again in a second. You would kind of like, you would like scrape out, oh, it's grass. We use grass, like dark green grass, and we like scrape it out. Then we mix it with some ethanol, and then we evaporate the ethanol, and then rehydrate the green stuff. We basically just make like a little really dark like ink, like a green ink, and then we place it. Let's do it again. We place it. It's hard to show you the very beginning. So here, so it starts off as just a little blob, and then when you put the plate in the solvent, it soaks from the bottom to the top. And as it does that, it pulls the different pigments to different distances.
Like
Because of the density.
Correct.
if this is in the paper question
Mhm.
test, if we're like asked to identify the pigment, are we supposed to remember the RF value, or it will give the pigment at the right to compare with?
Yeah, so you don't need to memorize any RF values. You don't even need to know how to calculate RF value. That's something that they ask you to do as part of the experiment, but it's actually not a learning objective. So it's a skill that it's something we would do. So for example, you would measure from the starting point and how far they went, and then just compare it to a table. So I imagine, I haven't seen this question in an exam yet, but I imagine they would probably have like a table of values and pigments, and then maybe like a blank, do you know what I mean? Like a,
Oh, okay. So they will supply you for comparison.
everything you need, basically. Yeah. Yeah.
So
But
just
it is a
testing
pretty
how
cool
you can
experiment.
read.
Sorry?
The test will basically be how you can read and match.
Yeah. Yeah, essentially. Yeah. Kind of like, DNA fingerprinting. It looks complicated, but it's really easy. You're just like matching like, oh, these lines are the same. Therefore, this is the father or whatever that thing. So yeah, this is one that we did. We would have normally done something very similar to this or exactly like this.
This looks like a grade 8 class.
No, that's 2017 class. I can't recognize anyone except Bjorn. I don't know if you guys remember Bjorn.
Dior?
Bjorn. Do you remember Bjorn Tin? And Jody, his sister was called Jody. And their dad worked at the school. Brian Tin. Yeah, anyway, it's not really important. Okay, so wait, where's the PPT? Oh no, what happened? Oh, here we go. So that would have been the lab that we would have done. So again, I really recommend click on that link on iLearn and just click through the activity so you can get an idea of how it goes. There's like a little quiz at the end as well for you to try. But let's move on. The rest of what we will do is objective. The first is to see the different factors and how they affect photosynthesis. And the second part is, again, very practical. It is like designing an experiment of how to measure photosynthesis, which you're already doing in a design for enzymes. So the basic idea is the same, but we're going to learn some ways if you were doing a plant-related experiment and wanted to measure photosynthesis. What are some of the ways you can do that? Okay, so two things, factors and what we know are called limiting factors. This should be reviewed from IGCSE, definitely. And then how to measure photosynthesis is more than anything, it's like common sense. So let's begin. We're gonna see 3 graphs, and these are graphs that either should be, you should recognize them when you see them and say, oh, for this, okay. Or if not, you want to be able to explain what the graph is doing. So in each of these graphs, what is happening is we are changing the x-axis, okay, down here. So we're going to say that as you go from left to right, that whatever's down here is increasing. So in this case, light intensity is increasing as you go this direction. And when we say light intensity, we're not saying wavelength. We're saying like let's say just no color or white light, but like the strength of it, right? Like how strong is that white light is increasing as you go from left to right. And on the y-axis, what is it going to do to photosynthesis? So can someone take a guess? What do we want to say for this orange line as far as how does light intensity increasing affect the rate of photosynthesis? What should we say here?
Excusez-moi.
How do you explain this section of the graph?
A positive relationship.
Okay, so that's the description of what's happening, yes. How can we explain why that happens?
As the light intensity increases, more light are used, are available for the plant to use.
So more light means more, what's the key term?
Energy.
More light means more.
Jensen said it.
What did you say?
Energy.
Energy, right, more energy. So from little to, some, the more light that is provided, the more energy there is, therefore the more photosynthesis that can be done. Now, we have an issue here. What's going on there?
Plateau.
What has happened beyond this point? Yes, plateau means flattening out, right? Well, what's the explanation we can give?
Has reached its maximum, right?
Optimum light intensity.
Okay, so optimum light intensity we would say happens like so somewhere here, right? This is your optimum light intensity. Beyond that doesn't matter, right? It's not necessary. But what has actually, like, how do we explain why? Why doesn't it keep increasing as you give it more powerful light?
Because all the chlorophylls are like already working at, like they're all working.
Yeah,
I mean, there's no more.
Could be. Maybe they're all working as hard as they can. There's not enough chlorophyll to take in the additional light that's being given. Sure, that could be one.
There's other factors now limiting.
There's other factors at play. So one thing we need to keep in mind is when we look at one of these graphs, we assume that everything else is the same, right? So there's the same amount of CO2, the same level of temperature, same level of humidity. The only thing that we're changing in order to get this graph is the light. So then we get to this idea of a limiting factor, right? A limiting factor is what's holding this back? What is holding this graph from continuing to go up higher this way? Well, what's holding it back is something else that we didn't allow to get more, right? Or to increase. So maybe it doesn't have enough CO2, or it doesn't have enough chloroplasts to take in the light, or it's not warm enough, whatever the case may be. Something else aside from light has become limiting. It is holding back the ability of this plant to do photosynthesis any higher than, like Joseph said, the optimum value.
So a limiting factor graph will look like
Well, if you had, I think there's still, at some point you will come to a limit. But yes, you should, for example, if you found out, oh, if I use these conditions, whatever they are, X amount of CO2, X amount of chloroplast, X amount of temperature, and then you get the graph we're looking at, and you say, what if I change X to X plus amount of CO2? Well, maybe you might get a little bit higher before it starts to plateau, right? Then you say, oh, maybe I change the, X temperature to X plus temperature with the X plus CO2, and then you get even higher the next time.
So what is the limiting factor?
And you wouldn't know until you tried another experiment where you changed one of them, right? So you can't actually know from just looking at this graph which one it is.
Okay.
You can only know by adjusting it and seeing if it changes things.
Okay.
Yeah, that's a good point. So you can, from a single graph, you cannot derive the limiting factor just like that. It's not possible. Clear enough? So main things are like these descriptions, right? How come it increases up to here? How come it plateaus beyond that?
Wait, Mr. Nilan.
Mm-hmm.
On the PPT, it says, therefore, light intensity is not the limiting factor.
Right.
But then on Google, it says the major limiting factors for photosynthesis are light, temperature, and carbon dioxide.
Yes, because in the next graphs, you'll see if we change this to something else, then light might become a limiting factor, right? Let's check out the next graph, and then I think that'll make sense. Okay, so if we look at the next graph here, so now we've changed this to not instead of light intensity, instead of light intensity, we're looking at carbon dioxide. So carbon dioxide from low to high. And in this case, light is the same. So we do not allow light intensity to change. We choose one intensity and it stays the same for all of this range. So in this situation, you could explain it the same way, right? Why does it increase from here to here? Well, CO2 is needed, right? It's the thing we need to make the glucose. So if you have a little bit of CO2, you can only do a little bit of photosynthesis. When you get more and more CO2, you can do more and more photosynthesis, right? But
so is it safe to say limiting factor varies with every situation?
Exactly.
I mean, graph.
Absolutely. Absolutely. So then if we try to explain why does it flatten out here, now instead of saying CO, because in the other one CO2 was limiting, in this case it's not, because we're adding more and more CO2, nothing's happening. Something else must be limiting it. Something else is holding it back. This time it could be light, because in this graph we haven't changed light. Okay, so the question is, what if we made the light more intense? Would we see a higher, higher curve before it starts to plateau? All right, so when carbon dioxide is increased, photosynthesis increases. So until this point, it is limiting, right? So if you don't give it enough, you're never going to reach as good of a photosynthesis as you can get. But then at a certain point, something different will become the limiting factor.
Nice.
Right? And this is another, so like why does adding more and more carbon dioxide not give you more higher rate of photosynthesis? Could be similar to what Aditi said, like even though you add more, like it has to happen in the chloroplast. If you only have so many chloroplasts, then you can only do photosynthesis at a certain level, right? It's the same idea as the enzyme argument, right? Enzyme, like the toilet analogy. If you only have so many toilets, only so many people can go at once, and you can't make that go any faster or different.
Okay.
Okay. So these two graphs are nearly identical for basically the same reasons. You just flip the what could be a limiting factor. And then we have one more graph. And this graph is not the same shape as these two, but it looks a lot like something we have seen like a million times in our lives. And that is this. Okay. Where have we seen this curve before?
Enzyme activity.
pH.
Enzyme and
pH.
pH.
Level.
No, not pH.
Denaturation.
Yeah, denaturation, but enzyme and what though?
Protein.
This thing, right? Enzyme and temperature. Enzyme and temperature. I know it looks very curved, like down. I would like for it to be like bam, but it looks a lot like enzyme with temperature. Slow increase. What's the slow increase about? Why does this happen?
Of energy.
Yeah. How do we describe it? What's happening?
They're having more kinetic energy.
More kinetic energy, therefore more
collision.
collisions in the correct orientation, right? So because, in this case, because photosynthesis is an enzyme like dependent reaction, right? You need enzymes for photosynthesis to happen, just like the majority of other reactions. So since photosynthesis is depending on enzymes, then the same effect that temperature has on any other enzyme, that same effect is going to be seen in photosynthesis, because the temperature affects the enzymes needed for photosynthesis. Therefore, if you mess up the enzymes, you're messing up the photosynthesis. Okay, so just like the enzyme idea, more kinetic energy, more collisions, more fitting of the substrate into the active site in the correct orientation. But we find the optimum, and then yes, denaturation, thank you. The active site is messed up, and therefore you can't do any of the reactions. If you can't do the reactions, you can't make the products of photosynthesis. Blam.
Okay.
Easy enough, right?
Yes.
A lot of repeats, a lot of stuff that comes back and forth. Okay.
Mr. Nealon,
Yeah.
does humidity cause a change in rate of photosynthesis? Because it affects rate of transpiration, right? But does transpiration affect rate of synthesis? That's a better question.
That is a really good question. Could be a really great research question, I wonder. I think there's probably an indirect link between the two. It would be, I think there'd be a few things. I think it would be dependent on the type of plant and like what its origins are. Because
the rainforest has like humid and it's like a lot.
Right, exactly. So I can't say for sure. I think it would have to be very extreme. Like it would have to be like either in very, very, very low humidity is it impacted. So like when you really reduce, when you really increase transpiration, or the opposite, when you really hinder transpiration. Because the idea is there needs to be water there, right? There has to be water at the leaf for it to happen. So if you affect the ability for water to be there, there must be some impact on photosynthesis too.
So like if you maximize all these factors, like temperature, CO2, and the other one, does it like rate of transpiration will then affect, right? Because if you if you use up a lot of water and water's not being pumped out fast enough,
Right.
then it will potentially affect like humid areas compared to like dry areas.
Yeah, I would think so. Yeah.
Okay, I see.
Okay, so the last part of this is just going to look at, this is kind of like one of those, again, normally if we were in the lab or something, we would kind of explore and look at a bunch of like equipment that we had, because when we get to it in the fall, and you start thinking about your IIs, and what kind of experiment you might want to do for your bio II. A lot of people will look at plants just because they're easy to control, and although they take a little bit of time, if you're like growing your own seedlings or you're germinating or whatever, you can do, let me tell you the benefits. One, you can do many trials without like a lot of stress. So you can just like plant like dozens of plants and monitor them all at once, or you can do repeats quite easily. The second thing is they don't take too much like care, right? You maybe check on them once a day or once every 2 days. And the last thing is it's very reliable, because there are so many different ways that you can collect data, or so many types of data you can collect using a plant. Okay, so what I'm going to go through is, or actually, even before we go through it, I'm just going to ask you. Let's try and think of some things. You think of a plant, okay? If you were to measure, like, what is the effect of blank on photosynthesis in this plant? What kinds of things would you try to measure in a plant that could tell you, like, what does rate of photosynthesis mean? Like, how would you know if a plant is photosynthesizing a lot or not a lot? What can a plant do that could tell you this?
How sweet it is.
Fruit.
One at a time.
Size.
Size. Very simple. Size. If it grows big, it's probably photosynthesizing more, right? Sure. Okay. What else?
The air is clean.
If the
air surrounding it becomes clean.
becomes clean. Can you be more measurable?
Like it feels different when you're staying in your room and when you're staying in a forest.
But could you measure that exactly? And how could you measure it? I know what you're trying to say, but if you were going to put it on a graph, what would you put on the graph?
API of
happiness.
You could do AQI, maybe. It's a bit indirect, but maybe. What are you really meaning? The exact thing that you are saying that the plant does.
Oxygen concentration?
Say again?
Oxygen concentration?
Yeah. Oxygen production, right? That's what you mean. So you could maybe measure how much oxygen it's producing. And you could argue that more oxygen produced means more photosynthesis. Sure. What else can we look at?
the glucose concentration or how sweet the plant might be after.
Okay. Yes, I would say glucose concentration could work. Sweet could be a little bit ambiguous, but glucose concentration, definitely you could do. It's maybe not so easy in our lab, but you could do, like, you could even do an iodine test, right? You could do the starch test, because if it produces glucose, then it's also producing starch, right? And if it's storing starch, or if it's storing more and more starch, then that would imply higher rates of photosynthesis. So what you could do, whereas in grade 9, like grade 9, we just did like blue, black, or orange, right? It's basically just yes or no. But in this case, we have a little bit more equipment that we can use. You could even do like a colorimeter, and you could do the iodine test and have like a range, like really dark, purple, black, and a range of like lighter towards orange, and say that like, oh, the darker ones are a lot of starch, therefore more photosynthesis. The lighter ones are less starch, therefore less photosynthesis. So that's a cool way to actually quantify starch production relating to photosynthesis. Okay, I'm trying to think, is there anything else that we haven't said? I think there's one. Joseph said size. So size could be, what could we be looking at in terms of size?
Leaf size.
Sure.
Also, maybe like the height.
Sure.
If it's a tree.
Yeah, could do plant heights. You can even do seedling heights, right? If you wanted to. Is there something a bit more, it's a bit tough to do, but when we're talking about size, what are we really meaning? Like what's happening to the glucose that's being used, and the starch and the cellulose?
Maybe the number of leaves.
Could do number of leaves. Yeah. How do you do, if you were talking about animals or humans, what's the equivalent to say if someone, say again?
Mass.
Right.
Like,
Right, mass. Now, what's the problem with a plant and its mass? What's a kind of...
You gotta find the biomass, right? The dry mass.
Exactly, the dry mass, which is, the biomass means dry mass, meaning you actually have to take out all the water, because the water contributes most of the mass of a plant. So what you do is you would grow your plant in whatever the condition, and then you basically take it out of the soil and put it in the oven, so all the water comes out, and then you take that mass, and that would be what's called the biomass. So I think we've gotten everything. There might be one that might be a bit tougher to get to, but let's see if we can just check out some of the ways that you could measure photosynthesis. So one is oxygen production. This is one we did in grade 9. We fake did it, right? Elodia, underwater, counting bubbles, right? Oxygen production is an indicator, an indirect indication of photosynthesis. If you wanted to do it a little bit more scientifically, instead of counting bubbles and sitting there for hours, you can put like a gas syringe and make it sealed so that the syringe actually moves, and then you can measure like an actual number for the volume of gas. Alternately, we actually have equipment in the lab, like an oxygen probe. So it's like you could put your plant in a bottle, and then you put this probe that plugs into your computer, and it's like a sensor, like a temperature sensor, or any kind of sensor, and it just detects how much oxygen is in the bottle, and you can leave it for a certain amount of time, and it'll collect data over however long you want it to, and then you suddenly have all these numbers, and it's like magic. It's brilliant. And we have quite a few of those in our lab. So again, like, keep the wheels turning, keep these ideas in your mind. So that's one. I should mention, the reason they have this equation here is because you want to see like a start and an end, right? So like if you want to rate of photosynthesis, you want to see how much photosynthesis is it doing over a period of time, right? That's usually a better indicator than just saying like X amount of photosynthesis can be tough. This is very weird, as you can see by this picture, is CO2 uptake. So CO2 uptake would be like, how much CO2 is it using? In this situation, you would have to know like how much CO2 did it start with, and then you have to like really have the plant like closed in an area. You can see how crazy this is. Like they put like a box around a part of this tree, like a leaf, and so they have a sensor plugged into that little box, and they're measuring the air around that leaf to say like, okay, when we first closed the box, there was this much CO2, and it's feeding data to a computer, right? And then over this amount of time, how does the amount of CO2 in this sealed box go down, which can let us come up with a rate. Right. If you're doing like an aquatic plant, it could be different. You could just measure the CO2 in the solution in the water around the plant, right? So that would be pH, because CO2 affects pH. Okay, here's our biomass one. So you could do dry mass, like we said. You could do iodine solution and do the starch test, just like we did with food tests in grade 9, or the fancy way by using a colorimeter.
How do we get the biomass? And that would just kill the plant?
Yeah, I mean, it would be the end of your experiment. So like your experiment would have it like grow it for X amount of days and then take the biomass.
But how do we know the initial biomass?
That is the problem. So you would have to have a control group, and then that's the only way is to compare it to a control group as opposed to a rate change. Right?
No.
Like, or you take the biomass of the same plants under similar conditions, and you take the mass by stopping it at certain times. You're making kind of a really big assumption in that case, because you're saying all of these seeds are identical, which is fine if you have large enough sample size. But let's say you stop one after 1 day and take the biomass. Then the next one you let it grow for 2 days, take that biomass. Next one for 3 days, 4 days, 5 days. And then you could connect those dots. Maybe that could work. That's a lot of work though. Okay. And what else? Okay, and then these are just guidance for just like your lab that you're doing, right? So it's saying, oh, if you're designing a photosynthesis lab, what kinds of things would you need to do? I'm not going to go through all of this stuff, but yeah. Oh, I forgot this slide. This is like one of those things they just sneak in there for fun. Changes the Earth's atmosphere due to photosynthesis. So they're saying that, like, remember when we said the Earth was a primordial soup and there was nothing alive? It was just like gases and rocks and lightning and volcanoes and all this stuff.
Yes.
What it's saying is, like, if you compare the atmosphere at that time to the atmosphere now, this is like where we started, right? It's like none of life existed without photosynthesis. Well, this is proof that that is the case, because at that time there was almost no gas in the atmosphere, right? But since then, you have a significant rise to the amount of oxygen that we know we have in the atmosphere today. And this is, you can trace that back to when photosynthesis started creating oxygen in the process of photolysis.
It's starting to make me feel that the primordial soup idea was developed by IB. They made and they're trying to convince us it's true.
They really want it to be the thing that you believe. It's like if suddenly you saw a lot of things about like creationism. They're like, and as we all know, God created the earth, right, guys? Yes.
And here is the proof why God made the earth, because blah 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 from this theory.
And it would come up in like cells topic and like evolution topic and
like every topic just sneak in one message.
Exactly. They really like the primordial earth theory, I suppose. Yes. But I think it's also because that is the currently best accepted theory. So that's why I said we would be able to finish. Look at that, we're done.
That's barely 1 hour.
Yeah, man. So SLs, as you
Bye.
said, you know, it's not too much that's new. It's basically, you know, similar stuff to what we've seen before. Yeah. Does anyone have any clarifying they would like from me?
Is I do Friday morning or Friday night?
Let's say night.
11:55 or
Sure.
59?
That's fine. Basically, how about this? I'm going to stop this recording for a second here.